Why do some people have allergies and others do not? And is it possible to eliminate allergies altogether? At Immunomic Therapeutics, we're developing a groundbreaking technology designed to stop the allergic response at its source. But first, let's take a look at the underlying biology of allergies. An allergy is an inappropriate immune response to normally harmless proteins called allergens, which are found in ragweed, peanuts, shellfish, or pet dander. During an allergic reaction, certain cells in our immune system, called dendritic cells, recognize these allergens and start a cascade of events that result in inflammation. The dendritic cells travel to our lymph nodes and interact with other cells of the immune system called Th2 cells. This culminates with the release of a type of antibody called IgE from immune cells called B cells. People with allergies have inappropriately high levels of IgE that circulate in the body and could make contact with allergy-causing proteins. Upon contacting the allergen protein again, the IgE causes our immune system to release chemical signals responsible for the symptoms of allergies, including histamines, prostaglandins, and leukotrienes. At Immunomic Therapeutics, we are approaching treating allergies in a completely different way. We're using a new type of DNA vaccine that gives our immune system the DNA instructions to encode allergen proteins as a way to address the biology behind the allergy. Immunomic Therapeutics' LAMP DNA vaccine therapy includes a short genetic sequence for lysosomal-associated membrane glycoproteins, or LAMP for short. Including the LAMP sequence allows our immune system to better process the DNA sequence. The DNA that encodes the allergen fused to the LAMP DNA is injected into the skin and taken up by dendritic cells. The immune system treats this foreign DNA like an infection and releases a chemical signal called interferon gamma. As the dendritic cells travel to the lymph nodes, they begin to manufacture the allergen LAMP DNA into a protein. The protein is synthesized and directly transported to a compartment inside the cell called the Golgi. Inside the Golgi, the antigen protein is modified with protective sugars and uses the LAMP signal to find other key immune system proteins, like the MHC-II protein that belongs in the lysosome. As these proteins all come together, they bud from the Golgi to form new lysosomes. Inside the highly acidic lysosome, the allergen protein is degraded into shorter pieces that are recognized by a special groove on the MHC-II molecules. The lysosome then moves to the surface or membrane of the dendritic cells, exposing the MHC-II and the antigen fragments to nearby patroller cells of the immune system, called Th1 cells. The Th1 cells are different from the Th2 cells responsible for allergies. Th1 cells cause B cells to release a type of antibody called IgG. Over time, the production of Th1 cells reprograms the body's response to an allergen by gradually eliminating the Th2 cells as well as the allergic response. Now when the body encounters this antigen protein, it rapidly disposes of it, and the allergy is no more.